Hello, everybody. Thank you for coming. For those of you I haven't met, I'm Mike Clegg. I'm Dean of the school, and I want to thank you for coming to this Dean's Lecture. And this is going to be a historic one because this is the coldest day in which we've ever had a Dean's Lecture, probably. And we're especially appreciative that Jean traveled all the way from Zimbabwe to be here to do this and brought her parents down. We want to welcome you to today's session. So just a little bit of background. These are talks that we invite people who have been appointed to or promoted to the rank of professor to come and talk, and they can talk about their work or really almost any aspect of their career that they want. And we do this because we want to take time to acknowledge what an important milestone this is in someone's career. It's very hard to get on the faculty here at Johns Hopkins, but it's really hard to get promoted to professor. And the decision is made, ostensibly by the advisory board of the school and the committee and then on to the board of trustees. But it's really, the decision is really made by colleagues from around the world, people who are professors at other universities or distinguished researchers who review the person's work and then send us letters and tell us whether what they think of the person's accomplishments. So it's a great honor to be promoted to the rank of professor. And I especially am so happy that we're celebrating you today, Jean. I remember the first time I met her in my office and things were falling apart in Zimbabwe, and I was so impressed with her passion and commitment to the people and the children of Zimbabwe. And I'm proud of our faculty and our alumni, what they go out and accomplish. But we're especially proud of you because you've done it in very difficult situations and with such a, as I said, a keen passion and commitment to the very vulnerable population. So I'll just tell you a little bit about her history. She's a Boilermaker, graduated from Purdue, got her MSPH from UCLA, and then her Doctor of Science from our school. And she began her career in 1987 at the Dana Center, headed by Al Sommer. Over in the Wilmer. And she started working on vitamin A at that time under Keith West, who's here, and of course Al. And Al is sorry he's not able to be here today. He was called on relatively short notice to go to the World Economic Forum in Davos to talk, which, as you know, he loves to do there. So but he's sorry he can't be here. And then after she got her doctorate, she came on the faculty here in the Department of International Health in the Division of Human Nutrition. In 1996, she moved to Zimbabwe to do the first trial of vitamin A in Africa, the Zvitambo vitamin A trial, which looked at the effect of vitamin A on maternal and on infant health and postpartum mother-to-child HIV transmission. She enrolled 14,000 mother-infant pairs, and about 5,000 of the mothers were HIV positive, making it the largest mother-to-child transmission trial in Africa. As you probably know, if you follow the vitamin A story, that showed no effect of vitamin A. And subsequently, and as she said to me, that was really disappointing. And other studies have shown that in Africa as well. But it provided the strongest evidence that exclusive breastfeeding was associated with reduced risk of postnatal HIV transmission. So as she continued to work in Zimbabwe, she kept that name of Zvitambo originally for the trial, and evolved into a multidisciplinary institute, NGO, that conducts research and provides broad technical support across the facets of maternal and infant health. It now employs 150 people, and they collaborate with investigators from 6 universities around the world, and which I really admire and like, as long as we keep the F&A, and provides international, that's an inside joke, that's about money, and provides international public health research experiences from students from our school, the University of Zimbabwe, and other institutions. And what she's going to tell us about today is her hypothesis that environmental enteropathy is a primary underlying cause of stunting of small height in children, and it can be prevented by protecting infants from fecal ingestion. So Zvitambo is now in the midst of a randomized trial, a community-based trial to test this hypothesis, and we're going to hear about it today. So Jean, congratulations, and thank you for coming.
Thank you very much, Dean Clegg. That was a very nice introduction. Thank you.
It's easy to give you a nice introduction.
Oh, thank you. So just to review, stunting is short stature for age. These two boys are the same age, but one is remarkably too short. It affects now a quarter of children in developing countries. It happens all between conception out here and 24 months of age. So this is height for age Z-score. And you can see that throughout the developing world, and particularly Asia and Africa in green and red, kids are born at about minus 5.5 Z-scores, and then it just plummets to at least minus 2 by 24 months. Oops. Consequences of stunting have been very motivating, and this is why people have been very interested in this. It causes about 20% of the under 5 mortality in children. And because linear growth parallels brain growth, children who are stunted at 2 years of age attend less school, perform more poorly while they're there, and have permanent cognitive deficits or lower IQs. And they grow into adults who earn less money and are more likely to have stunted children themselves. And then really poignantly, are at higher risk of what we usually think of the diseases of the wealthy: obesity, hypertension, and heart disease. So given that, the World Health Organization in 2012 made a resolution that the world would reduce the number of stunted children by 40% by the year 2025. So the goal was to go from 165 million to only 100 million by 2025. Now, the good news is there's been a dramatic decline in stunting globally, both in prevalence and in absolute numbers. So the proportion has dropped from 40% in 1990, when the Millennium Development Goals were first established, to about 26% now. And the numbers have fallen a lot too, from 250 million down to 165. This goal at 100 million is we're probably a little bit off. We're probably a little off that goal, but still quite close. If this is the goal, and this is the estimated if we carry on at the rate that we've been reducing up till now. The other good news is that there's unprecedented attention, and I really can't remember a time in my career in nutrition when there's been so much eagerness and political goodwill and energy and money going into undernutrition. So certainly the 2008 Lancet series on undernutrition it really got this going by quantifying the prevalence and the consequences and calling for us to focus on the first 1,000 days. At the same year, the Copenhagen consensus said that nutrition interventions were a good investment. They were amongst the most cost-effective investments for development. And almost all major donors and all the bilateral donors have now produced a nutrition strategy. And spending on nutrition-specific assistance has gone up by 60% since 2008, a period of time when many other costs have been cut. So what's driving this attention? Well, it's the clear evidence that stunting during the first 1,000 days costs us billions and billions of dollars in lost economic growth. And this is a recent analysis by the economist John Hadnot, that an adult who is stunted at 3 years has 66% less capita consumption or permanent income as an adult. And he estimated that for every dollar we invest in reducing stunting, it would generate $18 in economic growth. So here's the bad news. The bad news is Africa, where prevalence, this blue, is almost stagnant. It has dropped a little bit from 40% down to 30%. But because of population growth, the number of stunted children who live in Africa has actually increased from 46 million to 56 million. The worst bad news is that we don't know how to fix stunting. And you might say, well, isn't the answer obvious? Shouldn't we just feed them more? And this was a review done by Kay Dewey, where she reviewed 42 nutrition programs and research studies to look overall at what the effect of these interventions was on height for age Z-score. And the average one, the effect was 0.2 to 0.5, and the very best one was 0.7 of a Z-score. And this is against a deficit of -2. So at the very best, diet will be able to solve maybe a third of the problem. And I think it's very humbling for us in nutrition to know that there's never been a nutrition program or a research trial that has given an intervention and the intervention group ended up with an HAZ that was anywhere near to 0. The more recent Lancet series is a little bit more optimistic. They suggest that if we could implement the 10 best nutrition-specific or diet interventions with 90% coverage in the highest burden countries, we could reduce global stunting by 20%, or by, which is only half the goal of the World Health Organization. So right now, the best we know how to do this is to get down by 20%, and that's with spending huge amounts of money and really doing a really good job getting programs out. And we won't come close to meeting the goal of WHO. So then you say, well, isn't the answer obvious? If it's not nutrition, isn't it disease, and especially isn't it diarrhea? And diarrhea is certainly common during childhood, during the same period that stunting occurs, and contributes to poor growth. But it's been remarkably weak association with stunting. So this is the change in, this is from the Gambia, where they followed kids they've been following them for 6 decades there. And they were tracking sick clinic visits for diarrhea over between about 1980, 85, and 1993. And you can see that they'd done a lot of great things to reduce diarrhea prevalence in these villages. But over the same period of time, length for age at both 12 months of age and at 24 months of age did not budge. And it was that kind of data that caused the Lancet series in 2008 to suggest that even if we implemented sanitation and hygiene interventions with 99% coverage, and which we know will reduce diarrhea by 30%, these are very efficacious interventions for reducing diarrhea. But because this link is so weak, they estimated that the wash would only reduce stunting by about 2%. And that is because they assumed that the causal pathway between wash and reduced stunting would be mediated through reduced diarrhea. There are 2 studies that have both been done by people at Hopkins. Steve Esri, I don't know how many might know him, was an assistant professor when I was doing my coursework here. And he very tragically died of a brain tumor. But he was really one of the only people with a PhD in nutrition who was really on this connection of nutrition and water and sanitation. And he conducted a lot of cross-sectional analyses using DHS data. And what he noticed is that he would examine diarrhea, growth, across different levels of improved sanitation and water. And what he noticed is that the sanitation and water was more strongly associated with growth than it was with diarrhea. Similarly, Will Checkley, followed a cohort in Peru from birth to 24 months. At 24 months, the average child was 2.5 centimeters shorter than normal. 16% of that deficit could be explained by diarrhea, how much diarrhea the child had had, but 40% could be explained by the level of sanitation and water that the child had. And if we calculate, that's about 0.8 of a z-score. So this kind of an impact is similar to what the very best dietary interventions are able to provide. And it suggests that washes may be reducing stunting through a different pathway, and a pathway that's independent of diarrhea. At the same time, the Dunn Nutrition Center in the Gambia has been studying child growth and morbidity and diet for many, many years. And they had noticed over those years and reported that stunting was not associated with inadequate diet and wasn't corrected even with very good diet. And it was not associated with how many days the child was sick. But what it was associated with was gut permeability. So here's a growth chart for height for age and weight for age, and we see this common picture of it falling down over these weeks. So this was between 8 and 15 months of, 2 and 15 months of life. And just as the height for age is falling, the lactulose mannitol test, which is still the one good test we have of gut permeability, was rising in a sort of a mirrored way. So this condition of this permeable gut is called tropical enteropathy or environmental enteropathy. And what it is basically a subclinical, flat, in other words, this is the EE sample here, and this is normal small intestinal sample. So the villi are gone, but probably even more important than the flatness is its permeability. It's also amazing to see how common it is. So when I started reviewing all the literature on this, and most of the literature is from the 50s, 60s, and 70s, and they must have had easy way to get permission to get biopsies on people, on otherwise healthy people then. And so they were all gut biopsy studies, and virtually 100% of otherwise healthy people living in developing countries had small bowel biopsies that were characteristic of EE. And in fact, the very first time this was reported in the 50s was from Thailand. And the author said, well, it must be the chilies that they're eating in their diet that's aggravating their gut wall. So microbial translocation is when this, because of the permeable gut, so that when there are breaks in this gut lining, the microbial products that are in the lumen of the gut can pass through into the bloodstream and cause a chronic immune activation, which is metabolically expensive and diverts nutrients from growth to infection fighting. The veterinarians were way ahead of us on this and had demonstrated this in chickens when they raised chickens in either clean, steam-cleaned cages or dirty chicks raised in regular cages with dust and dander. And they noticed that the clean chicks would grow better than the dirty chicks, but when they added antibiotics to the feed of the dirty chicks, they would grow as well as the clean chicks. Adding antibiotics did not improve the growth in the clean chicks, and the poor growth in the dirty chicks was accompanied by an elevated, in this case they used interleukin-1, an indicator of immune stress, which was not observed either in the clean chicks or the dirty chicks on antibiotics. And I think that what both of these together suggest is that, both Gambian infants and dirty chicks, they mount an immune response, a chronic immune response, when they're living in an incessant microbial challenge, which diverts whatever they are eating away from growth and toward infection fighting. Now, Al and Keith used to have this when we were with them. They had an iceberg that they used to illustrate the fact that xerophthalmia, which is the eye condition that causes blinding, in children is this little piece of vitamin A deficiency. But subclinical vitamin A deficiency is really what's driving the public health importance of vitamin A deficiency, because it affects such a huge proportion of people. So I borrowed that from them and then would propose that diarrhea is really the tip of the enteric disease iceberg, and that it's environmental enteropathy, this low-grade chronic infection that's chipping away at kids' ability to grow, that on a public health level is really driving the importance of gut disease. So in 2009, I published this little hypothesis paper. It was a viewpoint paper. It promoted sanitation, which I knew nothing about and had never had any degree in it. It had no data in the whole, no collected data in the whole thing. And most of it was just reminding people of things that had already been published long time ago. The long-standing observations were the antibiotics improving growth in chickens, the ubiquity of environmental enteropathy among people living in poor living conditions, and the Gambian studies. And I hypothesized in that paper that EE may in fact be the major cause of childhood stunting, and that it might be able to be reduced through a wash intervention that would prevent kids from ingesting any kind of feces. I also wrote in that paper that I hypothesized that the primary causal pathway between wash and stunting, or poor wash and stunting, is environmental enteropathy, not diarrhea, and that optimal infant feeding practices, together with good sanitation and hygiene, would have an additive effect on improved linear growth. So we wanted to test this hypothesis in a trial. And it took us 7 years to do the formative work and raise the money, and to answer these questions. What kind of intervention will achieve an optimal infant diet? What kind of intervention will truly interrupt fecal-oral transmission in infants and young children? How can we measure environmental enteropathy in otherwise healthy infants? And how can we raise the money to do the trial? And Jim Toast would get aggravated at this and say, this trial is the longest gestation of anything that I've ever been involved with, because we kept year after year after year not quite having the trial ready to start. So the IYCF and formative research showed, not surprisingly, that the main problem of infant diets was very poor dietary diversity, because of the idea that they thought babies can't eat foods that need chewing. And the main and most important message that was integrated into interventions is a baby can eat any food an adult can eat, as long as it's prepared for the baby. And so there was lots of demonstrations on how to prepare all different kinds of foods that were in the household for a baby. And this was done by a doctoral student of Rebecca Stoltz's from Cornell. And when she used the TIPS methodology, and after trying these interventions and using these messages, the baby's diets were absolutely adequate in all nutrients except iron and zinc. And I think that was particularly impressive because it was 2008 when there was hardly any food in Zimbabwe that year. It was the most food insecure year that we've ever had. NutriButter was decided on to close the iron and zinc gap and was highly acceptable in the population. We started with the WASH formative research, doing a lot of structured observation of someone sitting here for 6 hours from early in the morning and watching all of the WASH and sanitation related behaviors. And one of the things we learned is that sanitation in Zimbabwe is highly valued, and toilets are highly valued especially a Blair ventilated improved pit latrine, which is the standard latrine in Zimbabwe. Very common misconceptions. People believed that infant stools were less offensive and less harmful than adult stools. People seldom used soap in washing their hands and frequently fed cold leftover food to babies. So based on this initial formative work, we decided on an intervention that was largely conventional wash interventions. So we decided, okay, this is going to be what the wash intervention will include. We'll build a Blair ventilated improved pit latrine. We'll provide 2 of these tippy taps. We'll provide soap. And we also provide water guard after the baby is 6 months for drinking water sterilization. But I was worried about that. And I was worried because I kept thinking, babies can live in a sea of feces, but it's not fecal-oral transmission unless it gets in their mouth. And I wondered, how is the feces getting in their mouths? What are the pathways here? And thinking that maybe they might be different than for older people, because the main things most people put in their mouths are food and water. But infants who are breastfed get the large majority of their fluid and food by breast milk. So we sent out students to do a baby observation study of 20 babies, and they watched 20 babies for 6 hours and recorded everything that they put in their mouth over that period of time, and recorded how often it went into their mouth and whether it was visibly dirty when it went in. We then returned and collected samples of 2 kinds of potential vectors. The things that had gone into their mouth most frequently, and the things that were most dirty that went into their mouth, even if they only went in occasionally. So we learned that children put a lot of things in their mouth, and the most frequently mouthed things are their own 2 hands. On average, they had 38 times of putting their hands in their mouth in the 6 hours. 75% of the time they were visibly dirty, and never, except in a whole bath, was a baby's hands ever washed with soap and water. But the dirtiest things kids put in their mouth is dirt, and 3 ate an average of 11 bites. One sat and ate 14 mouthfuls of soil, and then the mother came rushing over with a glass of water to help him get it down. Whole chicken feces, picking up the whole thing and chomping it right down, and of course putting stones in their mouth. And we saw that babies are fed on usually the ground or a kitchen floor, and both of those areas were highly contaminated with E. coli. So by looking at how much of these different foods the kids had eaten, we were able to calculate sort of the average E. coli per serving size of each of these to get an idea which is the likely most important pathway. Very surprisingly, and unlike most of the literature, we found no contamination in the infant food samples of these babies. About half of the drinking water was contaminated with E. coli, but because kids drank such small volumes of it, it actually contributed a much lower amount of E. coli. But soil, particularly soil, in the wet shaded places where out of the hot sun and near the laundry and the dishes, which is where babies are near their mothers, had 2,100 E. coli coliform forming units per serving size, and the chicken feces were particularly toxic. So we thought, really we can't have kids eating dirt and chicken poop in this study because we won't be able to prove the hypothesis. We've got to be able to interrupt this behavior. And so we finally, after a lot of argument with people in the early child development sector and others, and a lot of formative research and talking with mothers, have decided to go with this thing. So this is a mat that we can get in Zimbabwe. It's made out of that kind of plastic woven material that mealie meal comes in, or rice comes in Asia. And this thing is going to be bought from Minnesota. It's the largest selling play yard in the world. We're getting it for a really good price because they're really excited about what we're doing with their playpens. And one of the reasons for deciding to go with this commercial thing in this study, we had worked on developing something that could be locally made and something that could be purchased in Zimbabwe. But the problem is that the baby equipment industry is so, in this country, full of laws that protect children, that we decided we would go with something that had already had all the stamps of approval from all the pediatric associations, and it had been in the market for 60 years and never withdrawn from the market. It's fairly small. And we had lots of conversation about this because there are guidelines in the ScD literature about how many square meters you should have in a play yard at a nursery school for every child and things like that. And we decided to keep it small because we want to encourage mothers to use it for brief periods of time. So it's not someplace where you park your child for the whole afternoon. It's someplace where you put your child, when I had my children, when the phone rings, and you know that you're going to have your full attention off watching the child. But so the messaging around this will have to be very careful around, this is where they go for a few minutes when you need to do something else and you can't really be watching them. And we'd like to suggest a new paradigm here for WASH, thinking about, I call it baby WASH. So this is protecting children in the first 1,000 days from feces. And the unique exposures that they have are nappies and laundry water, which is usually just thrown in the yard, this geophagia, as well as their dirty hands, which are never clean, and the protective play space to interrupt this geophagia. Okay, so those were the development of the two interventions. Then the third thing we needed to do before we could have this trial is to figure out how to measure environmental enteropathy. And here we've been blessed with the very good fortune of having Andy Prendergast join us 2 years ago, who's a pediatrician and PhD in immunology. And we together created what we thought was a hypothesized causal pathway to figure out what should be measured. So we knew that we wanted to have a measurement of intestinal inflammation and permeability, of microbial translocation, activation of the immune system, the innate immune system, which pro-inflammatory cytokines should be measured, and insulin-like growth factor 1, which is the main hormone regulating linear growth and is switched off by high levels of pro-inflammatory cytokines. So we selected these indicators. So here we're going to measure IGF-1, here IL-6, AGP, and CRP. And this is, we can't find any evidence that's ever been looked at for environmental enteropathy, but it's widely used in the intestinal, the IBD literature, the, what's that? Thank you, the inflammatory bowel disease literature for children. And this is fatty acid binding protein, and I'll show you what that looks like in just a minute. So one of the good things that came out of the vitamin A trial that had negative effects, showing negative effects of vitamin A, is that we have a very large archive of over 400,000 specimens of these babies. And so we were able to harvest this to learn more about environmental enteropathy. In that trial, the 14,000 mothers, there were 9,500 who were HIV negative. And we went down to those babies at 18 months and selected 100 who had been stunted at 18 months and 100 who were not stunted at 18 months. And then we went back and pulled out all their blood samples at every 3-monthly visit, and as well looked at what their growth had done over that time. So the first thing that you see is that kids that are stunted at 18 months are already stunted at birth. They were on average already -1 height for age Z-score at birth. And then they slowly, over time, got even further, their growth velocity slowed, and they were even further and further from normal. So we start at the bottom of that causal mechanism, and we started by measuring this insulin-like growth factor. And what we found is that at every time point, from 6 weeks to 18 months, 6 weeks to 12 months, insulin-like growth factor was significantly and substantially higher in the non-stunted children than it was in the stunted children. And what we see, and we'll see now in several pieces of data on these children, is that something happens between 12 and 18 months. We see big differences in a lot of these indicators at 12 months that completely go away by 18 months. It does make us worry a bit if we really have 1,000 days. It may be something less than that. We then went on to measure the pro-inflammatory cytokines and found again that both CRP and AGP were higher at every time point, AGP from birth to 12 months and CRP from 6 weeks to 12 months. And again, you see at 18 months, this actually reversed and this gap closed again. There was no significant difference between stunted and non-stunted children for the other inflammatory markers that we measured. We then looked at the relationship between those inflammatory markers and insulin-like growth factor, and at every age there was a strong association between the two. So this is just an example at the 3-month age time point. And we see these very strong associations between insulin-like growth factor and CRP, AGP, soluble CD14, and interleukin-6. Finally, IFAP is a little protein that's mostly expressed in the tips of the small intestinal villi. And it's very sensitive to early bowel disease and early bowel inflammation. And we saw again, it doesn't look as different on this, but higher levels of IFAP in the stunted children compared to the non-stunted children. But interestingly, very high levels in both compared to European children. So healthy European children have IFAP levels down here between 20 and 183. Our non-stunted were way up over 1,000, and the stunted way up over 1,500. In fact, even European children with celiac disease only run up to about 800 picomoles per mil. So it suggests that all of these children in Zimbabwe had evidence of a lot of intestinal inflammation and damage compared with European children. The other nice thing, though, was that it looked like there was some repair going on between 12 and 18 months. We then ran models adjusting for birth weight, birth length, gender, maternal education, height, and MUAC. And the three markers that significantly predicted stunting at 18 months was insulin-like growth factor, CRP, and AGP. IFAP no longer did in this. It didn't predict stunting in a adjusted model. Finally, because we saw that things were changing so early in these children, we went back and pulled out the maternal plasma specimens from the Zvitambo archive and measured the insulin-like growth factor in mothers of the stunted children and the non-stunted children. And you can see that the mothers of stunted children had lower levels of insulin growth factor than the mothers of non-stunted children. And this is important because this hormone is important for fetal growth as well as for postnatal growth. So here are some take-home messages. This paper is in press, by the way. Prendergast is the first author. It's in press in PLOS ONE. Here's some take-home messages. Stunting at 18 months is associated with lower maternal IGF-1 at delivery and with lower infant IGF-1 throughout all of infancy. And it's associated with increased infant inflammation from 6 to 12 months. And the inflammation in babies is strongly associated with their levels of IGF-1 over the course of infancy. This IFAP, this measure of gut damage, is very high in both cases and controls, greater than children in the UK with inflammatory bowel disease, but always higher in cases than controls, although in adjusted analysis it didn't independently predict stunting. So we need some more work to figure that out. We added two more outcomes to the SHINE study before we finally got it designed, because we had gotten so interested in this role of pro-inflammatory cytokines and the fact that they seem to be switching off insulin-like growth factor. And only a few years before, in 2000 to 2001, this hormone had been described, hepcidin, as probably the major regulator of red blood cell production. And if anybody knows, Becky Stoltzfus is an iron guru, and so she was the one that was really keeping us up to date and working on this part of the study. So we added anemia as an outcome, as a primary outcome to the trial, hypothesizing that the chronic immune activation of environmental enteropathy would increase hepcidin, which reduces iron mobilization from stores and iron absorption and causes anemia. And all the nutrition people know how big both of these problems are and how overlapping they are in infants in developing countries. This is a word that I learned from Andy. It reminds me of, I'm going to date myself, but Mary Poppins, supercalifragilisticexpialidocious. It's a word that makes you feel smart when you learn how to say it. It's immunosenescence. I said, wow, what's that, Andy? He said, it's aging of the T cells. And it's lots of work. I see Diane in the audience, lots of work on this with HIV. That when T cells are constantly activated, they wear out and they don't work as well. And T cells that have seen a measles vaccine or they've seen a flu virus before don't recognize it when it comes around again. And we think that this immunosenescence, together with reduced absorption because of the gut damage, may be part of the story for impaired response to vaccines, especially oral vaccines. And we think we're going to be just very lucky that Zimbabwe is going to introduce rotavirus this year, just as our babies are getting, the babies in the trial are getting to the age where they would be eligible for it. And we are already drawing blood at the right time to be able to look at this. Also, because of these very early changes that we saw in stunting, in that case control study, we started thinking about going even back further to pregnancy and thinking about the mothers of these babies who have grown up in these same unhygienic, unsanitary conditions, and might not they have EE, which may be causing a constant chronic inflammation. And lots of studies show that inflammation reduces fetal growth. And so we are also hypothesizing that the severity of maternal EE will be associated with preterm birth, miscarriages, and preeclampsia. And you'll see when I show you the design of the trial, because we're enrolling mothers at 10 to 12 weeks of gestation, we have we're tracking and we're seeing a lot of these adverse effects and tracking them pretty accurately. So this is the design of the study. It's called SHINE, Sanitation, Hygiene, Infant Nutrition Efficacy Project. The key question is, are the independent and combined effects of a package of interventions to improve WASH and a packet of interventions to improve IYCF when initiated early in pregnancy and continued through 18 months postpartum, what is their impact on stunting and anemia? And you'll notice here that we state HIV negative and positive women. We're enrolling all women, but testing and identifying those who are HIV positive to be able to analyze them separately. Questions particularly around environmental enteropathy. What is the prevalence of environmental enteropathy amongst antenatal women? Does WASH reduce environmental enteropathy in babies? What's the relative importance of environmental enteropathy versus diarrhea in mediating the effects of improved WASH on reduced stunting? And finally, is maternal EE associated with adverse birth outcomes? And I want to just note that the literature suggests that adults with EE, it takes years of living in a clean place before their EE begins to heal and resolve. So we're not expecting mothers in the WASH arm, we're not expecting to change their EE. So this is really just an observational study that's nested within the trial. We also have a lot of implementation science nested into the trial, given all the behaviors that we're trying to get people to adopt. This is Mbuya, who finished his PhD at Cornell 5 years ago and chose to come back and join us. And he had focused his studies at Cornell on using village health workers, community health workers, to deliver nutrition messages. And so he's been the one behind all of the intervention development and now behind all of the implementation science. So we're trying to measure very carefully the whole black box of intervention fidelity, uptake, and impact. So it's a 2x2 factorial design trial of infant diet and improved WASH. We're enrolling all mothers who become newly pregnant who live in one of two districts almost right in the middle of Zimbabwe. So the two districts have a total population of about 180,000. We need 4,800 mothers in the study. And we predict that we should get that, we had initially thought we could get that in about 15 months. But it looks like it's going to take us more like 2 years to enroll those. We designed or defined the clusters for this trial as the catchment area served by a village health worker, or in some cases 3 village health workers if they work together as a team. And we wanted the arms to be balanced on several factors, including, really importantly, access to water and sanitation coverage. So in order to be able to do this, we had to measure the cluster-specific access to water and sanitation. So this is Robert Antizini, who's really quite a genius and designed this whole water survey. And we physically inspected all 6,000 water sources and tracked them for functionality, seasonality, and plotted their GIS coordinates on the map. Then we used Google Earth to actually, this is Google Earth with administrative boundary layover, and you get right down, and somebody actually spent 6 months counting all the households and getting the coordinates for that, and then adding that to the same map. It allowed us then to see where this triangle is a functional water source, and this is 500 meters, and that's 1,000 meters, to see how many houses, so these are all houses that are outside water coverage. And it allowed us to look at the distribution of access to water, and then to have an access to water measurement for every cluster. And in the randomization scheme, we've used both the ones that have very good access and the proportion that have very poor access. We also did a sanitation survey to look at sanitation coverage. Again, randomly selected households that were selected to yield cluster-specific estimates of sanitation coverage. Okay, so we ran, this is a special method, highly constrained randomization, which allowed us to yield treatment arms that were balanced not only on water and sanitation, but on a host of variables. So it allowed us to balance those 4 treatment arms on water, sanitation, geography, all of these things. So Larry Moulton did this, and I asked him, well, how are we going to do this? So he said, well, this is how you do it. You get the fastest computer you can find and run it for a week. So that's what he did. And then we called together all the 55 elected councilors from the 2 districts. And actually, I want to just point out that this is Larry at that randomization wearing a suit and tie. And what we then did is, once he had run the thousands of permutations that would that would meet the criteria of balance across the arms, he produced 10 maps that were randomly selected from all the ones that met the criteria. And we printed out great big posters of these maps, and we had 10 of them, randomization 1, 2, 3, up to 10. And we gave those out to all the leaders so that they could see where their district, where their people were, and what color it was. And that in map 1, it might be orange, and in map 2, it was green, and so forth. So this would show that if map 1 was chosen, if you lived in this district, you were in a blue arm, and if you lived in map 2 was chosen, you were in a yellow arm. So this was done in 2 stages with the leaders of these districts. So the first part was to randomly select one of the 10 maps. So that was done by me holding a plastic trash bag and somebody pulling out a ball that had the answer on it. We just had a ball labeled 1 to 10. And that was number 1, map number 1. So we quickly ran around and took away everybody's maps, 2 to 10. So they were only left with map number 1 in their lap. And then we did a second stage of allocating the 4 colors on the map. So all that had done is say which of these randomization maps we were going to use. And then we did the 4 colors on the map to which of the 4 treatment groups they would be allocated. And that was again by one leader pulling out a ball that said the wash arm, and another one pulling out a ball and said purple. And then they were put together. And of course, the goal of all this was to have a very transparent randomization scheme. I was scared to death to do this, but Larry pulled it off. To implement this trial, we're recruiting newly pregnant women by prospective pregnancy surveillance. So we've trained 360 village health workers to go around and visit all women between 15 and 49 every 5 weeks and ask them if they've had a menstrual period since the last visit. And if they haven't, offer them a urine pregnancy test. And then then they're referred to Shine using an SMS. All the village health workers have a cell phone. All of the interventions, the behavior change interventions, are delivered by the community level of the Ministry of Health. So it's the lowest cadre of Ministry of Health staff, village health workers. Just to acknowledge, this is Naomi Tavengwa, and she manages this whole process of training all of these village health workers and making sure that they deliver the modules that they're supposed to the right houses. Outcomes are measured by a separate group of people, 43 research nurses who ride motorbikes. And these are the 4 interventions: a standard of care, which still has a lot of good primary health care strengthened to it. And when we finish the study, everyone in this arm will get a Blair ventilated improved pit latrine. Infant nutrition is the messages we learned from the formative research, plus 20 grams of NutriButter every day between 6 and 18 months. The sanitation and hygiene is the latrine, 2 tippy taps with water, a soap, a water guard, play space, and the messages. And then there's an arm that gets all those interventions together. The interventions were formed into 5 modules for each intervention. So there's 5 modules for the WASH and 5 modules for the IYCF. And each of them include interactive tools and activities to make it fun for the mother to learn. So this is a flip chart in the IYCF intervention on how to prepare green leafy vegetables and increase nutrient density of porridge for the baby. This is a photo flip chart showing the mother who goes to the toilet, has germs on her hands, does not wash her hands, gets it on the food, and then she hands it to her baby, and there's the child eating this contaminated food. And then there's a similar flip chart with a woman who comes out of the bathroom and washes her hands, and there's no purple on the maize that goes into the kid's mouth. Oh, there it is. Then we have, this is the shit and ladders game. And so each one of these messages, each one of these are familiar pictures. So they were the pictures that were in the game or the tool that were used in the intervention. And this is a reinforcement module at the end. And so the families sit around and play this game with a village health worker. And then, of course, there's an opportunity for reinforcement of all the messages. I want to just end the talk today with some toilet stories. Just a minute, let me get some water. This has been the hardest thing I've ever tried to do, is this study. This is just, the logistics have just about killed us. And this guy is Peter Morgan. He's an old friend of Clive Schiff's, and they're sort of of the same era. They were both at the Research Institute in Zimbabwe at the same time. And just this last August, Peter was awarded the Stockholm Water Prize for his career and lifetime contributions to water and sanitation. So he invented the Blair ventilated improved pit latrine, which is the most widely exported health thing from Zimbabwe, and they're all over Africa. He also invented the bush pipe, and is still inventing. He's 71 years old now, and his laboratory is in his backyard. So if you go there, there are 15 or 20 different kinds of latrines and another 10 or 15 different kinds of pumps. So we had been told when we first started designing this that there's no way Zvitambo could build these latrines. You're a research institute, you do maternal child health research, you need to subcontract this to an NGO that knows how to do this. So we said, okay. So we interviewed so many NGOs, and we finally selected one who had an excellent reputation. And this is what happened when we started recruiting women into the trial. In January, they started out a little slow. April, pretty slow. June, not much faster. By June, I was panicked. I said, we've got to fix this. What's going on? And we had intense, long meetings, high-level meetings with all the directors of that organization. They said, yep, we're going to fix it. We're going to turn it around. Everything will be fine. Well, they didn't. And by August, we were into really deep problem because we had babies starting to be born. This was 9 months after we'd started recruiting, and those fetuses were on a conveyor belt to delivery. And basically, we were going to have 118 babies delivered in August, and every month thereafter, another 100 babies would get delivered. And Larry said, I think that we're going to have to exclude from the primary analysis any baby who doesn't get the latrine and the tippy tops before they're born. So I thought, oh, this is a real disaster. I wrote to Jim and Larry and Becky and told everybody what a crisis we were in. And of course, once again, Larry Moulton had the most helpful advice. He said, that is a problem. No toilets, no toilet trial. So I thought, yeah, that's true. So we had a toilet blitz this past fall. We fired that NGO. We learned how to build toilets. We built almost 800 of them between September and November. And then in December, we relaxed just a little bit until January, when they started falling in. And these were two that fell in the same month, and the rains were getting very torrential, and these had both been built by that NGO. So I called up Peter Morgan and I said, we've got to go down there, you've got to tell me why these latrines are falling in like this. So there's Peter down there, and he has a lot of them. He brings a digital camera and then sticks it into the squat hole and takes a photo in there with the flash going off so he can see what's going on in the inside. And he determined that he thought it was a perfect storm, that this NGO had used very poor quality bricks, that we'd had particularly harsh rain, and that these particular households were in relatively low-lying places, and that they had built these incredibly heavy latrines. So it was very heavy on a high water table, very loose sandy soil, and the things were just going down. So we had to write these up as SAEs and send them in to Joan, and I was so glad that nobody was in them when they came down, because then that really would have been an SAE, that she might have shut us down. And then I asked Peter to redesign, and so he went back out in his backyard and he redesigned a new latrine just for us, a revision of the Blair VIP design for very loose soils. Our timeline is that we've enrolled about 2,100 mothers, and we expect that we'll finish enrollment by the end of the year and complete field work by the end of 2016, and hopefully get the reports and analysis out by the end of 2017. And I want to certainly be very grateful for all of our support from the Bill and Melinda Gates Foundation and DFID. DFID were the first people that bought in on this and took a chance and invested in this idea, and smaller grants from other research organizations. So thank you.
Thank you, Jan. I'm sure lots of people have questions. I'm going to use the microphones because it's being recorded. So I'm going to go first. So why is it called the Blair VIP and not the Morgan VIP?
He's the humblest guy. And Dr. Blair was the head of the Research Institute at the time, and it was Dr. Blair's idea. Peter was actually trained as a marine biologist, and why he came to a landlocked country, I don't really know. But he did, and joined this Research Institute, and Dr. Blair said, why don't you do something useful and invent a latrine? And so he did, and then he named it after Dr. Blair.
It was a compliment, I guess.
So here's a VIP lecture.
It is.
So I'll just ask one more or less. So those slides you showed look just like celiac disease. So is this sprue? Is this what we used to call tropical sprue?
It's different than sprue because it's not symptomatic. And what's questionable in the literature is whether it's on the same, if it's part of the spectrum of the same disease, or whether it's a completely different disease, and people have different opinions about that.
Yeah, it looks just like sprue.
Yeah.
So I was happy. I don't know if Larry Moulton's here. I was happy when you showed Jim's picture. I thought, oh, it's too bad Jim's missing this, so I'm glad to see he's here. Oh, it's not working? Oh, yeah, there we go. So. Still not working. Oh, there we go. So maybe, Larry, since you were invoked so many times during this talk, it's such a guru, do you want to say anything?
We now have a trial.
But just on the sprue issue, though, but celiac disease is primarily an autoimmune response, right? This you don't think is autoimmune.
So there's celiac disease is an autoimmune disease that's found in developed countries, and it's called non-tropical sprue because it looks just like tropical sprue.
So tropical sprue is not an auto. It's not.
I don't know much about it. I don't know what the mechanism is, but it looks like Perul does. Or these guys.
So thank you, Jean. That was wonderful. And I just wanted to ask you about the 1,000 days, which you mentioned. And you mentioned measuring IGF-1 in the moms at delivery. Can you tell a little bit about the condition at birth? So what is the birth weight in this population? How many babies are born low birth weight, SGA, preterm? Because some of the work that we've been doing recently is linking the first part of the 1,000 days, which is the pregnancy period, to the later stunting. And you're alluding to some of that, but I wanted to get a better picture on what the situation. And maternal stunting, how common is that in this population?
Yeah, and I can't answer all of those right now. The maternal IGF her IGF level was related to her height. And I don't know exactly what that means yet. There's really very little IGF-1 data in the literature except on kids that aren't growing well in the United States and Europe. So there isn't a lot of data to really know what all those values mean. And certainly in SHINE, we want to look at IGF earlier in pregnancy and be able to see what's happening as early as the 10-week gestation visit. Now you were asking me about preterm and birth weight. So in the Zvitambo study, the mean birth weight was 3 kilos. And I think there, maybe Joanne remembers, because I just sent all this data.
About 3,000 grams on average.
And do you remember what the
And
preterm?
SGA, well, you excluded in that study people under 1,500.
Yeah.
So it's a little odd. And that was Ferrari. This is
Yeah,
more rural, right?
Yeah.
So,
I think the point is that I don't know if there's a lot of evidence of fetal growth restriction in this population. And I also wanted to get a sense of maternal undernutrition as a contributing factor.
well, I think in terms of the fetal, I think there's fetal stunting, probably not a lot of low birth weight, but fetal stunting. And
there is, but it's not nearly what it is in South Asia.
that's true.
That's the difference, I think.
Yep, that's true.
So basically, the deficits, it seems to start very early, almost at birth or soon after. So what's known about the microbiome of these two different kinds of children who either become stunted or don't become stunted?
We hope to learn. And so we have a collaborator at the University of British Columbia, and she's resubmitted her grant about 8 times, and I think next time I'll be surely successful. And we'll be able to look at the microbiome. Look at the microbiome. So we're collecting all the feces for her and be able to look at the microbiome of the mothers and the babies, and we'll look at mother-baby pairs and see how they're different, see whether they're different between stunted and non-stunted, and between whether the wash intervention changes that.
Okay, so let's make this our last question.
Is breastfeeding intentionally left out of the study, or is it going to get into the study at some later time?
No, it's a big part of the study. It's a standard of care intervention. So it's across all 4 intervention arms. And we've worked really hard to try to understand how to get exclusive breastfeeding rates up, because they're very low, like 5% exclusive breastfeeding to 6 months. So Amy Desai, actually, who was a doctoral student from here last year, did some qualitative work to try to understand what were the barriers of exclusive breastfeeding very early. And we learned that about 40% of babies are fed something besides breast milk in the first 2 weeks of life. But the mothers don't think of that thing as a food. It's a treatment for fontanelles or for colic. And the fontanelle in particular is this sort of mystical part of the body, and mothers have to be seen to be doing something about that fontanelle. So the treatment might be, even if they don't have a sunken fontanelle or a raised fontanelle, they'll be treating the baby for fontanelle, because this baby has an open fontanelle. And they put all kinds of different mixtures of local herbs and things in the baby's mouth. And
Right, because every color got it.
every color,
Every color got it.
gets 5 modules on exclusive breastfeeding. So I didn't say it.
Okay, so I see that Keith has grabbed the mic. So Keith, you'll be the last.
I just wanted to congratulate Eugene on a phenomenal talk and an even more phenomenal study. I don't think there's any of us know who is more perseverant in pulling off something like this. Question about the role, the intervention approaching women prior to and during the periconceptional period, because the stunting doesn't begin at birth; it begins at conception. What I didn't quite pick up what the intervention is for the mother to change her behavior through pregnancy in these 4 groups.
So in the wash arm, the two interventions which get delivered during pregnancy are the toilet and the hand washing and soap. We didn't want to introduce the water guard because we were worried that there might be confusion around exclusive breastfeeding. So we don't introduce the water guard until almost 6 months when the baby's ready to have water. And so one of the whole reasons for wanting to enroll them very early, in addition to being able to study the relationship between maternal EE and adverse birth outcomes, is to be able to give the family all that time to practice these new behaviors with the goal that by the time the wash baby shows up, that's a really clean house with everybody using the latrine and everybody washing their hands with soap. So the mother is the key recipient of those messages. There isn't an intervention for her. I'm not sure if you meant that. There's a lot of standard antenatal health care that we facilitate or provide, but in terms of a randomized intervention, there's nothing for the mother. But a lot of behaviors we're asking her to change in addition to everybody else in her household.
So there's lots of other questions I'm sure people have, but we'll ask everybody to go after. When you get the results of the trial.
Thank you. Thank you very much.
So,